Good morning, student. In my last video, I've explained and you also understood the structure of DNA. Now, we're going to complete that structure of DNA right now in this video. So, where we left earlier, from there I am going to start and to complete the whole process that we need to know related to the structure of DNA. Here, this is pentose sugar, deoxyribose sugar. It is united by nitrogenous base. Nitrogenous base, adenine, guanine, cytosine, thymine, and then it is called nucleoside. When it unites with a phosphoric acid, then it is called a nucleotide. Then again, we find that another sugar, deoxyribose sugar, uniting with nitrogenous base, nucleoside, then it unite with the phosphoric acid. Dear student, as you are knowing that a phosphoric acid is having 3 OH, as I told you earlier. So what is happening now? 1 OH of the phosphoric acid here unite with the OH of sugar, and then this OH moves out. A molecule of water moves out. Got it? And here also another OH. See, another OH of the phosphoric acid or phosphorus here. Here another OH unite with the OH of sugar. So here also one molecule of water goes out. So ultimately what you are finding? 2 molecules of water moves out, leads to CO bond. Okay, which is the linkage. The linkage is called the ester linkage. We're finding the occurrence of 2 ester linkage. Where? This one is one, this one is another. 2 means what? Di. So, it is called diester linkage. So, the diester linkage helps in the formation of polynucleotide. Like that, number of nucleotide unites together, there will be diester linkage to form it. Another thing, students, you have to keep in mind that the acidic condition of DNA is due to the presence of phosphoric acid. Okay? Now let's quick move to the structure. How the DNA are having the arrangement. Now see, as I told you, the DNA, this is the DNA, see student, and this DNA, it is double helix. So till now that I was telling you related to one polynucleotide, this is one polynucleotide. But in DNA, there is the occurrence of 2 polynucleotides, not one. These 2 polynucleotides are united by what? Hydrogen bonds. Now students, you have to keep in mind in case of the bonding, how that occurs. Here you see this is sugar. It unites with a nitrogenous base. Okay? It unites with what? Nitrogenous base. And this is phosphoric acid. Suppose this is this polynucleotide, one polynucleotide number 1. Now, there will be another polynucleotide. Let me put it with another color, red, so that you can understand it very clearly. This is another polynucleotide. This is the nitrogenous base, and here you are finding the phosphoric acid. So this is a nucleotide of one polynucleotide, and this is the nucleotide of polynucleotide number 2. Understood the thing? Polynucleotide number 2. Now what you are finding? The nucleotide which is going to unite with another nucleotide need to be complementary. What is the meaning of complementary? Need to be contemplating each other. Who decide how it will be a complementary? It is the nitrogenous base. How? If there is adenine. Okay? Purine group will be uniting with pyrimidines. So there, the nitrogenous base of the coming nucleotide to form a pairing with it need to be thymine. Okay? That means adenine always unite with whom? Thymine. Like that, guanine will be pairing or forming the complementary nucleotide with cytosine. Just you take the reverse now. Thymine will be pairing with whom? With adenine. And now I am putting here reverse order. Cytosine will pairing with whom? With guanine. Now student, you have to keep in mind another aspect. What is that? That hydrogen bond that is formed in the process of union of two polynucleotide chain. In case of joining of adenine with the thymine, there will be two hydrogen bond. Keep in mind. And in case of joining of guanine with that of the cytosine, there will be 3 hydrogen bond. So therefore it will be the same like that of the earlier, 2 hydrogen bond, and here how many? It will be 3 hydrogen bond. So these are how the bonding takes place. So what do you find? 2 polynucleotides unite with each other because of what? Hydrogen bond. Hydrogen bond. So that is the another important thing we need to know. Quickly we will be going for the structure to understand the final step of DNA structure. That means how their nucleotides and other aspects are arranged. So this is a double helix. It is called having 2 polynucleotides. So it is like a helix, double helix it is called. This DNA. Now, this DNA here, it completely rotates, making around 360 degree. Okay? And this is one turn. Understood? This is one turn. I am taking out this one from here. How it will be? It will be like this. This is one turn. Like this one. One turn. One turn of this one is 34 Armstrong. In some books you may find to be nm, nanometer, doesn't matter. So that is the unit by which it is measured. Now students, you have to keep in mind, in each turn, this is a turn, there is the occurrence of 10 nucleotides in each of the polynucleotide. That means it has 2 polynucleotides. Therefore, 10 complementary nucleotides will be there in each turn. So, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. So complementary will be 10 here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Understood? So definitely the consecutive nucleotide will have a distance of 3.4 Armstrong. Why? Because the length is 34 Armstrong and there are 10 nucleotides. So each two consecutive nucleotides will have 3.4 Armstrong. White. This is white. White is 20 Armstrong. Now see, this one moves anti-parallel. What is the meaning of anti-parallel? It moves anti-parallel. That means if you keep two person, if one head is in the north direction, another head is in the south direction, that is anti-parallel. So before anti-parallel, I will be telling you another thing. See here, the DNA, if it is attached here in the 3 dash, here it will be, this side will be 5 dash, and here will be 5 dash and 3 dash. Okay. So anti-parallel means here you see it is moving in the upward direction and here it will be moving in the downward direction. So these are the parallels, but the movement is anti-parallel. So DNA strands are having two polynucleotides which remain completely coiled with each other. So this is the structure and this is the arrangement of the DNA and the nucleotide that is present in it. Thank you, students.